Good evening, ladies and gentlemen. Welcome to day 2 of the Neurotransmitters educational series for week 6. This is our second last week. I thank you all who have logged on. Thank you for not being tired of the educational series. This week we are looking at Parkinson's. We had a very engaging and insightful introduction yesterday by Dr. Raymond Mugume, and he looked at diagnosis, introduction to Parkinson's disease, the definitions, and the epidemiology. So today, we are looking at etiology, the neuroscience, the neurobiology behind Parkinson's disease. I'm Dr. Kemirembe, a finalist at Makerere University, doing Master of Medicine in Psychiatry, and you're all very welcome. Since we intend to keep time, I welcome Dr. Mugume to get on with today's presentation. During the presentation, I'll share with you the link to the Neurotransmitters website where you'll find all these resources and more. Resources about the brain from the world's best researchers, the most up-to-date resources, videos, PowerPoint presentations, all you'll enjoy the website. I'll also share with you the recordings. We went online. We are now sharing the recordings on YouTube, and every session that we shall be having will be shared on YouTube. Thank you very much. I see Dr. Mugume is ready with his presentation. You're most welcome.
Good evening, colleagues. Welcome to yet the second day of Parkinson's disease. And today we'll be talking about neurobiology and the etiology of Parkinson's disease. Just a little recap. Yesterday we looked at how Parkinson came around, how the term, or who coined the term Parkinson's disease. We also looked at the 4 cardinal symptoms, which was bradykinesia, rigidity. We looked at the tremor, whether peer rolling or resting. And then we looked at the postural instability or gait impairment that usually comes in the late stages. And we also looked at how you can diagnose, which was you just need the bradykinesia and the other 3, any 2 of the other 3 that remain. But also you need to put into context. And so today we are going to talk about the neurobiology. How does Parkinson's disease come about? And before we do that, before we delve deep into that, we shall look at some normal physiology that we should keep in mind, and then we can relate better. So we both know that we have brains, and part of those brains that we need to keep in mind about is the motor cortex, which literally motor cortex is made of the primary motor cortex, which is literally controls your hands, your lips, your trunk muscles, your neck, your ears, and everything that is really in the upper part, but most especially the hands. And we shall see why we need to keep in mind of this. Then we have the premotor cortex, which is it controls everything else to do with how you move your facial muscles, but also it helps to initiate to help you to initiate movement and also inhibit movement, as we shall see. And then the supplementary motor area. And what we need to know about the motor cortex is that, of course, it is intertwined between the midbrain, the basal ganglia, and then the cortex. And so it's able to perform its activities. So there are neurons that are interconnected with those areas that I've talked about. And so that is what, importantly, we need to keep in mind. Of course, we need to talk about also the basal ganglia, which literally is the initiator of movements or the inhibitor of movement, depending where the innervation is coming from. And also this is also dependent, of course, on the neurotransmitter dopamine from the substantia nigra. And also what we need to know with the basal ganglia is that you have the putamen and the caudate nucleus. The putamen is responsible for motor movement. So you hear putamen, you know movement or motor movement. You hear the caudate nucleus, you think about the cognition. it just deals with the cognition. The globus pallidus, it does have innervation to the thalamus, which in turn has innervation to the cortex. So that is depending on whether it is the globus interna or the globus externa, as we shall see, movement can be initiated or it can be inhibited. Then we talk about the subthalamic nucleus, which we shall continue to understand better in the indirect pathway and how it also impacts the thalamus in inhibiting movement. Then the substantia nigra, we have the substantia nigra interna and the externa. I know the substantia nigra's pars compacta, which is of importance that we need to pay attention to. Depending on what neurons are innervating or are leaving, it can either be excitatory or inhibitory, depending on, again, on which receptors are also in play. And so let's have this in mind, but we shall understand it better. It also brings us to the direct and indirect motor pathways. So these are the two pathways that the direct is for, when you hear about the direct motor pathway, you know that you are going to initiate movement. The indirect pathway, it is to inhibit all the other motor movements that might happen in the absence of what? Of control, okay? So, and the working is really within the dopamine receptors mainly. And just to clarify, like here, just to give you the normal physiology, in the direct pathway, you have the cortex. The cortex is like the main controller, okay? It is the main controller to say, okay, you can make movement or you don't have to make movement. So this arrow usually, there are neurons that come from the cortex, which are usually excitatory with the main neurotransmitters, glutamine. They come from the cortex, they go to the striatum, and the striatum is both the putamen and the caudate. The putamen is mainly for motor, and then caudate nucleus, of course, there is cognition, where you have to negotiate, do I walk now or do I wait later? And then you have striatal neurons that that send inhibitory GABA neurotransmitter to the globus pallidum, the internal segment, which is the GPI, which will be known as the GPI, okay? And also there are neurons that also go to the substantia what? Nigra, corpus, the compactor division of it, which is this one, okay? And then, which are themselves also inhibitory to the thalamus. Well, in this diagram, we do not have the thalamus, but I will just take us back just to remember that we have our thalamus here, okay? You have our putamen and the caudate nucleus here. You have the substantia nigra, okay? So what I'm saying is that you have neurons that come to the striatum, this is the striatum. And usually those ones are excitatory. They have glutamine as the main excitatory neurotransmitter. And then neurons that come from this, from the striatum, usually go to the globus pallidum. So you have the globus pallidum, which is this one exactly. So you have the striatum, putamen and caudate. Then you have neurons that go to the globus pallidus, the internal segment, and also some go to the thalamus, okay? And in that, they are usually inhibitory to the thalamus. So they inhibit the thalamus in other words, okay? And this double inhibition from both the putamen as well as the globus pallidus internal segment usually allow the excitation of the cortex to allow the premotor cortex to initiate movements, of course. And so this is the direct pathway. It is really direct like that, okay? So this is what we see in this diagram, that you, from the cortex, you go to the striatum, where you have D1. D1 is usually for movement, you initiate movement, and D2 usually inhibitory, okay? So you have then the direct pathway, where you go to the globus pallidus internal segment, that in turn inhibits the thalamus, and then which allows the excitation of the cortex to initiate movement. That is in the direct pathway. And in the indirect pathway, you still have, in the indirect pathway, you also still start from the cortex, okay? And from the cortex, you still have the glutamate as the neurotransmitter. And then from the striatal neurons, they usually go to inhibit the globus pallidus external segment and also the subthalamic nucleus. Okay, so the globus pallidus external normally inhibits the subthalamic nucleus, as you can see in this arrow. And then when that happens, the subthalamic nucleus usually becomes more active, and then sends excitatory signals to both the globus pallidum internal segment as well as the substantia nigra reticulata, which in this it will be part of the substantia nigra, okay? And then from there we shall have, this will increase the inhibitory output of the thalamus, okay? Which again, the thalamus is here. So you have excitatory globus pallidus interna as well as the substantia nigra reticulata, which inhibits the thalamus and in turn inhibits the cortex. So that's how you inhibit the motor movements. In normal people, these two pathways are usually in check of each other, okay, depending on if you want to make movement or otherwise, okay? Then it brings us again back to the neurotransmitters. We know that we have a presynaptic membrane, presynaptic knob, where you have neurotransmitters kept in vesicles, and they're usually brought into the synapse, which is this space between the presynaptic and the postsynaptic membrane. This is the synapse, the cleft, okay? And then these neurotransmitters will usually attach on the postsynaptic membrane, and then depending whether second messenger, RNA, or maybe metabolic or otherwise, you can have postsynaptic changes happening, okay? So it is important also to have that in mind. So what is the role of dopamine? We know that dopamine, of course, plays a critical role, especially in control of movement. We have seen it is very important in both the direct and indirect pathway. And it can work on different receptors depending on if the receptor is excitatory or inhibitory. D1, excitatory, D2 is inhibitory. And what we need to know is that most of the dopaminergic neurons are in the substantia nigra, which is found in the midbrain. And this is what Charcot found out, that it is the most affected area in Parkinson's disease. And the pars compacta we have seen in the direct pathway, it does modulate the activity of the striatum as well as the reticulata. And this modulation is very important, okay? Let's look at the pathogenesis after understanding the physiology. So we have what we call the alpha-synuclein, which are usually misfolded proteins that are usually found at synapses. Their mechanism is not very clear, but the alpha-synuclein is usually soluble. It is non-folded in normal individuals, and it's usually found at the synapse, and so it's made of protein, what we need to know. But in disease, what we know now is that usually this protein, we do not know exactly the mechanism it aggregates. But in Parkinson's disease, after you have gotten loss of neurons, it kinds of folds and forms what we call fibrils that later form Lewy bodies, okay? So you have monomeric synuclein that usually aggregate, and the more they aggregate, they form fibrils, and these end up being Lewy bodies. And I remember I mentioned that in one of the cadavers that Charcot did discover, he found that there were Lewy bodies within the substantia nigra, okay? And also, most patients who will have aggregation of this alpha-synuclein is that they will have mutations in the alpha-synuclein gene on chromosome 4, the small arm, okay? So that is something we need to understand. And mostly in idiopathic Parkinson's disease, this makes the difference, okay? In terms of knowing which Parkinsonism the patient has. And it is starting to be an area of developing treatments in treatment of Parkinson's disease. Lewy bodies, like I said, I already explained to them, they're usually really aggregates of alpha-synuclein, and there's no consensus on how they develop or how they are related to the neurodegeneration of Parkinson's disease. But what we need to know is that they're actually made of alpha-synuclein, and we know that they also contribute to the cognitive changes that we see in Parkinson's disease as we shall go on. Neuronal cell death in the substantia nigra is reflected, or it has been known, and how these dopaminergic neurons get to degenerate. We shall get to know that, of course, there is mitochondrial dysfunction in individual cells, which later leaves us to have oxidative stress that only makes the vicious cycle into lobbing microglial cells and so on as we shall move forward. But it's really important that we understand that these neurons die within the substantia nigra. So we talked about the Braak staging of Parkinson's disease yesterday. We have stage 1. And stage 1, we said that the dorsal of the vagus nerve that usually innervates the GIT is usually affected. And between stage 1 and stage 2, what we know is that in these 2 stages, the alpha-synuclein aggregates have been more marked, okay? And so along the way, you have the Lewy bodies also forming, and we don't usually have a lot of motor symptoms, but we have more of psychiatric manifestations as we looked at sleep disturbances, depression, constipation, among others are more pronounced in this stage. But we do not have per se neuronal cell loss, except aggregation of alpha-synuclein in this. So again, this alpha-synuclein does not only aggregate in the brain, it also aggregates in the GIT, it aggregates in the skin, and there is also a school of thought of maybe in the prodromal phase of Parkinson's disease, we could actually do biopsies just to see if we have Lewy bodies. The moment you find Lewy bodies, especially in the GIT or in the enteric nervous system, maybe that's the time we need to start targeting or start treatment very early. But these are advanced schools of thoughts that we haven't related with, but they are happening elsewhere except Uganda yet. So we know that the enteric nervous system is also a brain of its own, and there's mounting evidence that the diseases that affect the enteric nervous system also affect the CNS. And in particular of this is that once there is accumulation of alpha-synuclein, and then there is dopamine also being affected in the GIT, there's propagation of these oxidants, of these inflammatory components to the brain, and so it kind of makes a vicious cycle. So it is the gut-brain axis comes into the picture. So as again, like I said, that maybe detection of the enteric nervous system pathology could therefore be potentially useful in monitoring and diagnosis of Parkinson's disease. So the moment you see patients with constipation, you're giving your normal remedies, it's not happening, the people are feeding well, please, you may need to think about if there's maybe either inflammation or there's in the long run, like 15 years later, you might have Parkinson's disease. So we have alpha-synuclein and the prion hypothesis. Not until today, I did not know that prion, prion is really stands for the proteinaceous infectious particle. And why is it coming into Parkinson's disease? It is now understood that these aggregates of misfolded proteins can actually propagate themselves from within the cell to the neighboring cells and ultimately to other areas of the brain. So it is almost in the same, using that same understanding, like we have seen in other diseases like the Creutzfeldt-Jakob syndrome, where you have prion disease. It is the same principle that alpha-synuclein, the misfolded proteins, also affect other cells as observed in the dorsal motor nucleus of the vagus nerve, and which later spread. So it starts from the GIT and then propagates upwards. And that is why we are talking about the prion disease phenomenon. And what we also need to know is that there was a study that was done. They transplanted fetal dopaminergic neurons in patients who had had loss of neurons in the substantia nigra. And because these patients already had Lewy body-like inclusions, they found that over time, after they had done the transplant, 11 to 16 years later, these patients continued to deteriorate. And they found that actually when they did the pathology, they found that even the grafts that they had given the patients had already been congested with the alpha-synuclein. So that's why we need to understand this hypothesis. Like I said, the proteinaceous infectious particles. So you have, these are the cells containing the normal population of properly folded proteins. Then one of the proteins, which could be the alpha-synuclein, forms, and then it ends up influencing all these other proteins to also fold, and so that's how it ends up propagating, and then the effect comes in. So how strong is the evidence that Parkinson's disease is a prion disease? Again, like I said, there have been experimental studies within the animal studies that have showed that actually there is usually cell-to-cell transfer of alpha-synuclein. And I talked about it yesterday in the level of pathology, that you have within the cell, then cell to cell, and ultimately the whole brain is involved, okay, from one region to the other. So that's how strong the evidence is. Because these neurons usually secrete the alpha-synuclein, then it is absorbed by the corresponding cells or the neighboring cells, and then the cycle goes on and on. So that is, so it has been thought that maybe because of this way, which was observed in other prion disorders like the sporadic Creutzfeldt-Jakob disease, it is the same way that they have found that this could be the same phenomenon, that the alpha-synuclein actually affects the neurons and the cells, okay, within the brain. So again, this is just to emphasize what I've just said. And it is also observed in Alzheimer's disease, which is also a neurodegenerative condition that is associated with dementia. And this is how the propagation also happens. What are some of the risk factors that we need to keep in mind? So we know that we have talked about genes. For Parkinson's disease, the only gene that has been implicated is the mutation of the alpha-synuclein gene on chromosome 4 that has been implicated. In fact, it is an autosomal recessive. It is an autosomal recessive. So you need both parents to have that autosomal recessive gene for you to have an etiology with Parkinson's, to have Parkinson's disease. And usually it will be very early, it progresses faster, and cognitive conditions are very prominent, okay? So you need the genes and in the environment to work together to give you an etiology of Parkinson's disease. And what we see in the pathogenesis is that you have neurons being excited very much. We have inflammation coming in. We have the mitochondria being dysfunctional, and then the oxidative stress increasing. And then this also affects the enzymes that usually, or the cells that usually come in to clear the debris, and then leading to cell death. That is what we need to understand. So this is a level of evidence on some of the things that we know that are risk factors. Pesticide, we know that most of our farmers that are exposed to pesticide usually are more at risk of getting this Parkinson's disease. And even in the labs, they use certain pesticides to destroy dopamine receptors within these animal models, okay? With weak evidence, we are not sure. It is just more like maybe there's correlation that maybe if you have a high milk consumption, especially in men, you could be at risk. Those who have high intake or have chronic anemia would be at risk of developing Parkinson's disease, but it is just weak evidence. We are not very certain. There is correlation, but we cannot say for sure that it is causation. Then for reduced risk, people who drink coffee at a reduced risk, those who have uric acids high, they also say that they are at a lower risk. Tobacco use, those who use tobacco in all forms, they say also they have a reduced risk. And with the weak evidence, if you do vitamin A intake, if you drink alcohol, drinking tea, these other teas, whether it is herbal tea or these aroma teas, vigorous physical activity, taking NSAIDs, they say they have a reduced risk, but we do not have enough evidence to support it, okay? Susceptibility genes. These are genes. When we talk about susceptibility genes, it means that you having that gene would increase your risk by 3 to 4 folds, but it does not mean that you having the gene necessarily means that you will get the disease, okay? Because then we have to think about the environment, if you're exposed to pesticides or not, and other risk factors that I just mentioned, okay? And in this case, these ones are just loci within the chromosomes that have been implicated, that if you have most of these loci within the chromosomes and their mutations, your risk is very high of getting Parkinson's disease. For autosomal recessive, you need both parents to have those autosomal recessive genes for you to be at risk of Parkinson's disease. For the autosomal dominant, especially like the pinky one, usually you need one parent to have it, and you'll be at risk of getting Parkinson's disease. The other one is the mutation on the alpha-synuclein mutation. You will be at risk. It is an autosomal dominant. The other one is the GBA1, which is Gabriel-Serudez gene, which is a gene in the lysosomes. When you have that, usually when you have that mutation, you are at an increased risk of getting Parkinson's disease, and it is mostly implicated in the Gaucher's disease, okay? So that is, there are a number of genes that really are being pointed out, but none has been identified specifically for Parkinson's disease, that once you have a mutation in this, then you will have Parkinson's disease right away, okay? And this is what I was saying, chromosome 14, you have a mutation in the alpha-synuclein, you will have early onset of Parkinson's disease, and it is a faster progression. So again, where you see AD, that is autosomal dominant, autosomal recessive, okay? Once here, you need one parent, you just need one copy of it and you will be at risk, okay? And the locus, of course, is PARK1 and PARK4 with point mutations and multiplications of these loci being passed on from your parents, and then Parkin and PINK1 and DJ1, these are all autosomal recessive. So you need both parents to have both of them for you to be at risk of getting Parkinson's disease. Then we have the leucine, the LRRK2, which is the leucine-rich repeat kinase 2. Which is also autosomal dominant. So the moment you have this, a parent has one, and so it becomes more like familial, okay? These are what we call like familial Parkinson's disease. You'll be able to have that if you can do some biomarkers and you identify these, or you do some DNA and you identify these, okay? Again, genetic factors in Parkinson's disease are varying by ethnicity. So what we need to know with this, with genetics, is that with gene-wide association studies, we now know that most of the studies have been done in a specific ethnic group. And so we cannot use that to determine that maybe in the African ethnicity, we shall have the same. Okay, so we cannot, like most of the studies we now know have been carried out in the European ethnicity or the Asian ethnicity. And we know that now in the current state that African ethnicity has variation in their gene wide association studies, okay? So that creates a good problem that then we might need maybe to do our own studies, or in the different, like using an example within East Africa, like we know that now, like in Eldoret, the gene-wide association studies say that they are too varying, like compared to anywhere in the world, okay? And then people who are in Uganda are different from those in the north and those in the western part of Uganda, okay? So that means that you cannot say that because of maybe a gene that was identified in the European ethnicity, if you find it within the Baganda here, then it means that they actually exposed, they are at a greater risk of getting what? Parkinson's disease. So that is the last statement, which is the disclaimer that I've just explained. The common pathways underlying Parkinson's pathogenesis is, we always have a proteosome, which is usually made of these, we have these genes that I've already talked about. Most of them are autosomal recessive, and they contribute into the accumulation of misfolded proteins, of course, with bearing in mind that you have a mutation probably here, okay? And this, in turn, oxidative stress can cause the accumulation, or vice versa. That is the double arrow, okay? Or the proteosome also can cause oxidative stress. Once that happens, it usually will lead to apoptosis, which is cell death. But also, the mitochondrial dysfunction within the mitochondria, especially the mitochondrial complexity 1 gene mutation, usually has been implicated to cause mitochondrial dysfunction in functionality. And we know that the mitochondria really is for energy. So once it's not working, there's no energy, and then it will lead to oxidative stress, okay? Then I talked about the GBA1, which is a gene within the lysosome that helps it to function and to do clearance of the cellular debris or let's say the unwanted cellular debris that needs to be cleared, or to formulation of, or to create space for more new regeneration of other neurons happening. But when you have that gene mutation, the lysosome is not able to do its job, and in a way, it is either eaten up, or it gets destroyed, or it contributes again to accumulation of this, okay? So you see, it is a cocktail of things that is happening while you're seeing the patient individually, but there are a lot of things that are happening at both the cellular level and also at the structure level. Again, this is just to emphasize that mitochondria plays an important central role in the pathogenesis, but it varies. The mitochondrial deficiency varies among patients, and the PARKIN1 and PINK1 proteins they do function in the mitochondria, but we do not know what exactly they do in the pathogenesis of Parkinson's disease, okay? That needs to be understood better. Again, this is just to emphasize what I already explained. And of course, how the mitochondrial dysfunction happens is you can have mutations in the mitochondrial DNA, which of course affects the functionality of the mitochondria, as we all know. And then in turn, it affects the whole process or the pathogenesis. So it contributes to the pathogenesis. We need to talk about So you know that Parkinson's disease happens in the elderly. And by the fact that they are elderly, they already have chronic inflammation, okay? So having chronic inflammation as well as having alpha-synuclein coming in, usually you would understand that in normal instances, the microglia will change shape and then take up roles of clearing, okay? They will release inflammatory mediators, which cause inflammation, and that lobbies the microphagia to come in and clear, okay? But in the elderly, because the system is now working almost below minimum, they already have chronic inflammation. And because there is the microglia will still be mobilized, the inflammatory markers will be released, okay? Or the mediators will be released, but then they will not be able to, they end up recruiting more leukocytes from the peripheral. And this usually will promote clearance of the cell debris. But in our case here in the elderly, these inflammatory mediators, because again, the cell cycle is not functioning properly, there is no like stop to the inflammation. It ends up also affecting the healthy neurons, and this contributes to the neurodegeneration that will happen in these patients, and then the progression of disease. So it is important to understand the neuroinflammation, and also to know that by the fact that our patients are already old, they already have chronic inflammation, it just the process increases the neurodegeneration of our neurons. Then, of course, I talked about the GBA1, which is the glucocerebrosidase. And in the gene, I talked about it, once it is affected, you have a predisposition of Parkinson's disease. Usually I saw one case when I was an undergrad. They usually have, they have a bad liver, their heart is compromised, because, lysosomes are everywhere in our bodies, okay? So imagine if they are not doing their job. Jaundice is high, the heart is compromised, they have cognitive issues, the child they don't survive really to see their 14th birthday, okay? So it is very important to know. And this is just to tell you more about the clinical features of those that were found with GBA1 positive and they developed Parkinson's disease. You can see that There's a symmetric onset, they have bradykinesia, but mostly the cognitive changes will be more pronounced as compared to these other resting tremor and rigidity that are telltale of Parkinson's disease. What is the future of Parkinson's disease? I think from this neurobiology, it humbles us to know that there are a lot of challenges in finding proper treatments or disease-modifying strategies for these neurodegenerative diseases. And also, it also makes us want to think that maybe these disease-modifying strategies, the ones that are discovered, maybe the ones that will work within the enteric system, the one that will work on the alpha-synuclein, maybe if it is either chelation, the ones that may be help in regeneration of dopamine or the dopaminergic neurons, they have to be, you have to use them in consensus, so you have to use them together. Like example, the example in cases maybe combining drugs with different mechanisms of action, which is usually routinely done like in cancer, but we don't see that in Parkinson's disease. And we also need to appreciate that Parkinson's disease is a collection of diseases, each with a different phenotype, and maybe it needs to be treated differently, okay? Because you've seen that someone can have Parkinson's disease because they have a mutation with the alpha-synuclein gene or with the GBA1 gene, or even all the other, the LRKK1 gene. So we need maybe to think differently when we are thinking about treatment modalities within Parkinson's disease. With that, I come to an end of my presentation. I'm open to the questions. Thank you very much. Back to you, moderator.
Thank you so much, Dr. Mugume, for dissecting this topic. It's not an easy one, but you've done us good. And I'm sure everyone who has attended is much better than when they came on. So I am not seeing a question on the chat. There was somebody requesting for the link to the recordings, and I've sent it. Please access the recordings on YouTube. And if once you subscribe, you'll be able to get every session that has been recorded. Please note that some sessions have not been recorded. So do not be surprised if there are some that you don't find. However, starting from last week, all the sessions that we've had and we shall be having henceforth will be recorded. I do not see any questions. Maybe if there's a quick one, you can raise your hand. Dr. Mugume, most of them are saying, well done, thank you. From Brian Wesige and from Dr. Ruta Chamira Dorena. Sunday has also appreciated. So really, we are grateful to all of you for attending. We know it's not easy with the end of year schedule, but please continue to come. We just have one more day tomorrow to look at the management. I know we are all very interested. Kindly look for colleagues who may also be interested and share the link with them so that they can join us tomorrow, and even remind them it will be our last session for the week, and then we shall have the very last session in the series, the 7th week. I also don't see the faculty. Dr. Mwesiga was on and he had to drop off for another meeting, but he was with us. Thanks once again. Unless somebody else has a question, I think I'll wish all of us a good night. A reminder that the Neurotorium website has all this information and more, so you are most welcome to check it out as well. Thank you very much and have a good night.